Today we're going to talk about sensory information. So we're going to learn about the olfaction, which is smelling, audition, which is auditory, sight, which is these little eyeballs, and taste, which is his little mouth. And all of our systems work very much like Max's, who's now giving me a little love bites because I woke him up. Okay, thank you. That's gross. Thank you. You're so sweet. Today's topic is sensation. So we're going to go through all the senses that we think of and that I showed you on the cat, but we're also going to go through some senses that you might not be familiar with. For example, equilibrium, that's going to be balance and body position. It uses your inner ear just like hearing does, except it uses different parts of the inner ear. Electroreception is the ability to sense electric fields around you. A lot of fish do this and sharks do this. And then magnetoreception, that's the picture I have here. That's the ability to sense the magnetic fields on Earth. For example, being able to tell where magnetic north is. And we think that turtles use magnetic fields to migrate. And so they can feel when they're pointed north, and so they know what direction they're going. We also have somatosensation. That's sensation that lets you feel your body. Your vestibular organs, that's the inner ear I just mentioned. That's going to be orientation and balance. It's going to be gravity, acceleration, deceleration. Then we have proprioception. Proprioception is the awareness of where your body is in space. So you have sensors in your muscles that feel when they are stretched or compressed. You have sensors in your joints that feel when they are stretched or compressed. So you know if your legs are bent without looking at them. We also have kinesthesia, which is the ability to know where your body is in orientation to itself without looking. So if your eyes closed laying in bed, you don't need to go and observe where your feet are to tell where your feet are. You have receptors for that, and your body is interpreting it constantly. Our first sense that we're going to get into is olfaction, your sense of smell. This is cool because it's pretty much a quick connection to your brain. Your olfaction use bipolar neurons. They're called bipolar because they have two extensions. One is dendrites, one's an axon, and the cell body is right in the middle. So it looks like it's got two arms, so bi for two. These bipolar neurons are in your olfactory cavity. So they're in your nasal cavity, and they extend through a bone right into your sinuses. So they're like up back here. They're not in your nose, they're up inside like the hole in your forehead. So if we were to look at the space on this skull, this is in and up. So it'd be like right between your eyeballs in your forehead. And the way these receptors work is they are chemical receptors. So for you to smell something like a bit of vanilla or chocolate or fish, a piece of that thing has to get up into your nasal cavity and it has to physically bind to the dendrites of this axon or this neuron. So it is chemically touching it. When it touches the dendrites, it creates a depolarization event, and that depolarization event runs up the neuron right to your olfactory bulb. So there's no intermediate, there's no relay. It just goes from the smell to your brain. The olfactory bulb is a projection of your brain. This is your brain right above it. It's connected to it. And so smell is often associated with really strong memories and really strong emotions. Also disgust. So if you think about smelling rotten milk or a rotten egg, your body knows really quickly that that's not something that you want to consume, and you're going to keep yourself safe from eating spoiled food. We have about 350 different olfactory receptors. With those, we can smell 10,000 different odors, and that's nothing compared to our other friends. So here we have a snake. These organisms and all vertebrates, except for us, they have an organ called the vomeronasal. You have a bone in your nasal cavity called the vomer. So name for the bone and the location. And this vomeronasal organ allows vertebrates to smell pheromones. Pheromones are hormones produced by other organisms that give them lots of information about that other organism. The way the snake smells is it extends its tongue, which is sticky, and in the air, it picks up all these particles and then it sucks it back in. It sucks the particles off it and it feeds them into its vomeronasal organ. And from there, it can piece apart what pheromones are there using these chemical receptors in its olfactory system. Cats have a really impressive one too. So here's a cat yawning, and we can see the awesome hard palate, but then there's this little round ball. That little round ball right behind the teeth, that is the vomeronasal organ. When cats smell a pheromone that they think is interesting and they want to smell it even more, they want to smell it harder, they do the stinky face. They make that little tongue out, derpy face that you see right there on the right. That's called the phlegm response. And the phlegm response isn't just for cats, although cats do it all the time. Other vertebrates do it as well. So they are intentionally opening their mouth and making this space to help move air across their vomeronasal organ so they can smell pheromones better. Dogs have an incredible sense of olfaction. In comparison to us, dogs' sense of smell is 1,000 to 100,000 times more sensitive. It varies dependent on breed, so there is a range. Their olfactory bulb is huge. Here is their brain. This is the brain stem. This is the cerebellum. This is the cerebrum. And then here, this massive piece in the beginning, that's the olfactory bulb. It's about 40 times larger than ours. They also have way more receptors in their nasal area for smelling. They have about 30 million, and their epithelial surface is 5 times larger than ours. Plus, they don't want to stop smelling or stop breathing when they want to do one or the other. Like we have to intentionally smell while we breathe. Dogs have 2 separate pathways. So they can smell something and breathe at the same time. There's 2 tracks. I've got a video up on Blackboard for you about how dogs smell, and it's pretty awesome. I can't wait for you to see it. Our next sense is taste. It's gustation. Gustation is also using chemical receptors. That means that the piece of thing that you're going to be tasting has to get in contact with the neuron that's going to taste it. Now, we have taste buds. Those are the bumps on your tongue, but they're not actually taste buds. So the bumps on your tongue are called papilla, and they have a lot of different functions. One of them is increasing surface area. Another one is for friction, so we're kind of creating an abrasive surface. But on those papilla, you have taste buds. So here is the tongue. This is the tip of the tongue that would be by the lips. This is the back by the throat. And we have all these different types of bumps. Those are the papilla. There's different papilla in different locations on the tongue. You can see these big ones, the circumvallate ones, when you open your mouth really wide, they're in the back there. If we look closely at the papilla, this is a picture from a microscope. You can see the whole papilla is this big structure, but this is not a taste bud. The taste buds are embedded in the sides. So here, this little portion right here, that's a taste bud. Right above it, that's a taste bud. Over here, taste buds in the sides. So right in the armpits, these little pink circles, those are the taste buds. So along the circumference of your taste bud, of your papilla, there are taste buds. Our 5 primary tastes are sour, sweet, salt, bitter, and umami, which tastes like meat or savory, mushroomy, rich, very deep earthy flavors. Here's another image of our lingual papilla, lingual for tongue, papilla being these bumps. So here are those big papilla in the back, and then we have different types of papilla all over the surface. This is another picture in the microscope of the edge of a papilla. So this is the edge of the papilla. You're looking down into this fold right here. And on the edge of this papilla, we can see the taste bud. Here's the taste bud. It's got a pore at the top. That's how the thing you're going to taste is going to get in there. And then inside this pore, that's where we have our sensory neurons. This image is drawn below. So this is a recreation of what we're seeing in the microscope. Here's your hole, the taste pore. And inside that pore, this whole structure is your taste bud. In your taste bud, you have your taste hairs, which are basically dendrites of these neurons. Those dendrites chemically bind to the corn you're eating or to the toast you had for breakfast, whatever it is. And that creates a depolarization event. Which taste buds are activated and how much they're activated, your brain interprets as flavor. So it's a chemical reaction that causes a depolarization event that goes up to your brain, and your brain makes sense of it. Our next sense is audition, which is hearing. Audition is neat because we are creating sound waves with our voices. Sound waves are little packets of pressure in the air. Those packets of pressure in the air are going to your ear. They're jiggling your eardrum, and they are creating pressure waves inside of a liquid in your inner ear. And that bends a membrane. And where the membrane bends, your brain interprets as pitch. So how high or low a sound is and how loud it is. That is going to be amplitude. The pitch of a sound, if it's deep or high pitched, is going to be determined by the frequency of these pressure waves. The frequency are how short the wavelengths are. So a long wavelength or short wavelength, which is the distance between two peaks, is the pitch. And then the amplitude is the volume. The amplitude is how high your wavelength goes. So this dotted line here that goes lower, that would be a quieter sound. This solid line here that goes higher, that would be a louder sound. Let's get into this. Our ear is divided into three parts. We have the external ear. The outer part's called the auricle. So that's the part that you can pierce. Then you have the ear canal, and that takes you to your eardrum, which is the tympanic membrane. Think timpani is a type of drum, so tympanic. This outer ear is important for capturing sound waves. And now we're going to transmit that sound wave into something your brain can understand. The middle ear is in this air space. So this is full of air. There's a canal that connects it down to your throat. So if you've ever gone up an elevation in a car and you've tried to pop your ears and you've held your nose and you blew back, you were forcing air up this tube called the eustachian tube, and you were equalizing pressure in your middle ear. So this is connected to the outside, the middle ear. When you have a sore throat, when you get sick and your ears start to hurt, it's often because this tube gets inflamed or filled with mucus, and then you can't equalize pressure very well. So this eustachian tube helps you control the pressure in your middle ear. Your middle ear is this air chamber, and it has three little bones in it. They're called ossicles because they're tiny little bones. They should be called like osciitas, but they're ossicles. So ossus is for bone. The first one is called the malleus. The malleus is right up against the tympanic membrane. Then you have the incus. That is going to connect the malleus to the next one called the stapes. The stapes fits into this place called the oval window. The oval window connects to the inner ear. Now, the stapes is like a plunger. It's up against a membrane. It's connected to it. And when your eardrum gets hit with sound waves, your eardrum vibrates in and out, and that creates movement in those little bones, and your stapes plunges in and out on the membrane of the oval window. That's going to transmit those pressure waves from the sound into the fluid in your inner ear. When you create those pressure waves, you're going to need somewhere for them to go. You can't put pressure into a closed liquid system. So here below, we have the round window, oval window, and round window based on shape. The round window has a membrane over it, and it's going to make this so you can see it. It's going to pop in and out with your pressure waves. So it's a little bit like pressure release, but it doesn't let the liquid go anywhere. But if we were to look at this round window, it would be blowing inward and outward with the pressure waves that are going through your middle ear. Your inner ear is really amazing. We're going to have some portions that help us feel gravity, and we're going to have some portions that help us feel acceleration. We'll talk about that. But we also have the cochlea. The cochlea is the part we're going to use for hearing. So let's get into the cochlea. The cochlea is a snail shell in shape. So it's a spiral getting smaller and smaller as it goes. And inside the cochlea, we're going to have liquid. The pressure waves from the oval window are going to move through the liquid, through the spiral, get to the end, spiral back out to the round window. So we're going to have a path in through the spiral and a path out through the spiral. As pressure waves move through the spiral, they're going to vibrate the membrane. When it vibrates the membrane, there's different areas that vibrate more heavily based on frequency. And that area is going to bend. And where it bends allows us to hear sound. So let me show you what this looks like. Let's orient ourselves. Here's a tuning fork. It's making a sound. Here are the pressure waves in the air. This is explaining the wavelength and sound relationship. We discussed that. Here's your outer ear, your auricle. Here's your ear canal, your auditory canal. This is your tympanic membrane. Your tympanic membrane gets hit by these airwaves. It's going to vibrate the little bones, the little osseouses. Those little bones are going to take your stapes. It's going to plunge in and out on your oval window. And as it plunges in and out on the oval window, it's going to send pressure waves through the cochlea. This below here is part of the cochlea. It's been straightened out so we can look at it, but this is happening in a spiral. Here is the pressure waves coming from the oval window, from the stapes. These pressure waves move through this top canal, which is called the scala vestibuli. They move all the way through and they come back out the scala tympani. As they move through, these pressure waves are going to create push downs in the membrane. And if they push down, it can be felt by this center canal. This is called the organ of Corti. And the whole canal itself is called the cochlear duct. So this is the duct inside the cochlea. We're going to zoom in on the organ of Corti in 1 second. Okay, now we have zoomed in and we are looking at the cochlea. Here is the cochlea. On the top, you have your vestibule, and then you have your other vestibule. So these are spaces. The top one is where the pressure waves go in. The bottom one is the tympani. That's where they come out. Now, inside, you have your cochlear duct. And inside the cochlear duct, you have your organ of Corti. This membrane here is the one that's going to wiggle with pressure waves. So the pressure waves are moving through the top duct. And through this top duct, you're going to have this membrane wiggle. It's going to wiggle in relation to the frequency of the pressure waves moving through. So it doesn't wiggle randomly. It wriggles at certain hertz. And it will push down into the cochlear duct. And as it pushes down into the cochlear duct, it's going to displace this liquid. And as it displaces this liquid, it's going to move this piece of tissue. This piece of tissue is called the tectorial membrane. And as the tectorial membrane moves, it's going to tap neurons below it. Here's a zoom in. This is the tectorial membrane. Here are hair cells. Those are a type of neuron. They have their little protrusions. Think about those as like dendrites sticking upward. And that tectorial membrane is going to push down and it's going to tap those hair cells. Every time it taps a hair cell, the hair cells bend, and that bending creates a depolarization event. That depolarization goes up to your brain, and your brain interprets it as sound. You hear sound because pressure waves tap a neuron. Isn't that wild? Let's get into the other parts of your inner ear. So we just used your cochlea. That allowed us to hear. This was audition. Now we're going to use our vestibule. So vestibule just means empty space. You can use vestibule for other places too. Inside the vestibule, we have two spots, the utricle and the saccule. Those are going to be used to detect gravity. Then we have three semicircular canals. These semicircular canals are going to be used to detect acceleration and deceleration. The semicircular canals are in our three axes for a three-dimensional space that we live in. So there's the x-axis, there's the y-axis, and then there's the z-axis. So each of these is oriented to feel motion in a different direction. Let's look at the semicircular canals first. Here's a person. This person is not moving. When you are not moving, there is no action in the neurons in your semicircular canals. These semicircular canals are filled with a fluid, and at the armpit of each of them, there is a structure called an ampulla. So here, this whole structure, inside the ampulla, there is a packet of gel called a cupula, and then there are hair cells embedded in the gel. These are your hair cells here. This whole structure, the hair cells and the gel, is called the crista ampullaris. So the liquid inside your semicircular canal is called endolymph. That's the same liquid that's in your cochlea. And then we have some supportive cells. So those are great too. But this is what's going to happen. When your person moves, they're going to spin their head right or left. Maybe they're going to twirl. He's doing ballet. When you move, the fluid in your semicircular canals moves as well. So here my endolymph is moving because I'm moving. As it moves, it bends that gel pad, it bends the cupula. As it bends the cupula, it bends the hair cells inside of it. And your body understands that as moving. Just like hair cells bending, your brain interpreted sound. Hair cells in a different location bending, your body interprets as movement, as spinning. If someone drinks too much alcohol, they often feel like they get the spins. That's because your endolymph becomes more fluid. And so a little movement that wouldn't normally make you feel like it's spinning will make your endolymph just like fly sideways and bend your hair cells more. And so your body interprets I'm spinning because you haven't really diluted it, but your hair cells are feeling more activity because of the alcohol. Now let's talk about how we detect gravity. So below our semicircular canals in our vestibule, in that space, you have your macula, which is the little space where we're going to have our sensation. And the two locations where macula are found are the utricle and the saccule. So here, this whole thing is the macula. Inside of this, we have a liquid, our endolymph. And then we're going to have a gel pad. Our gel pad this time is called an otolith. Otolith. I've heard it pronounced oolith or oolith. Those are fine too. Otolith is what I'm going to say. And in this gel pad, you have hair cells. Now, the gel pad isn't going to be moved by the endolymph for gravity. For us to detect gravity, it's going to be moved by little tiny crystals that are sitting on top of it. So here's a person, his head is upright, he's not feeling like he's tipping. And in his ear, we have little tiny calcium carbonate crystals sitting on top of the otolith, and no one is bending the hair cells. When our friend tips his head forward, those crystals are acted on by gravity. They move forward, like if you were to tip a bowl full of sand, it moves forward. And as those crystals move, they push the otolith with them, and that causes the hair cells to bend, and your brain interprets that as tipping. Isn't that wild? So when you tilt your head back and forth and you feel that motion, it's because you have little crystals that are bending neurons, and your brain interprets that as gravity. By the way, when you are falling, you feel that sensation of falling. That's because the little crystals lift as you fall, and your hair cells feel that they're not pushing on them as much. And your brain interprets that as a decrease in the weight of the crystals, and it recognizes, oh, I'm falling. The inner ear is so amazing. Your eye is equally as amazing. So to detect vision, to detect light, we are going to use specialized neurons called rods and cones. Light is actually packets of photons. Photons come from light sources, and that's what's going to be hitting these neurons, and we're going to be detecting those photons. Now, photons can come in different wavelengths, and we see different wavelengths as different colors. So shorter wavelengths are more purpley than longer wavelengths, which are more red. The wavelength determines the color. Rods are going to be used to see light in general. They just see photons. They're not good at picking up detail. They don't pick up color. These are the neurons that you use when you're out at twilight and everything starts to look grayscale, or what you use in your peripheral vision. So if I'm looking forward, everything I can see over here, my brain's kind of just making up. But I'm using my rods to see it, and my rods mostly just see light. Now your cones, they're called cones because they're kind of cone shaped. Your rod does not have a tapered tip. These cones, they're particular. They do pick up color. We're going to use them for our detailed vision, but we only have 3 types. We have blue, we have green, and we have red. And every color that we can see is some variation of those cones being activated. I have a video here for you on Blackboard that I want you to check out about how we see color. It's really interesting. Here's a detailed look at your eyeball. So this is the front of your face. The protective layer of your eyeball is called the cornea. Below the cornea, we have a little space. This is called the anterior chamber. The anterior chamber is part of the anterior cavity, and it is filled with a liquid called aqueous humor. Then you have your posterior chamber, and you have your iris. Your iris is the muscle that controls the amount of light that's going into your eyeball. That's also the piece of your eye that has color. So that is your iris. There's a hole in the center of your iris that looks black because there's no light inside your eyeball. So it looks black because we're looking into a black chamber. And that is called the pupil. The pupil is going to dilate and constrict because the iris is going to get bigger and smaller. It's going to open and close, and that's going to allow us to control the light into our eye. Behind the iris, where light goes in through the pupil, we have a lens. The lens is going to focus light onto our retina, which is where the neurons are. The retina is in the back of the eyeball, and the lens is going to change shape when we look at things that are close versus things that are far away. So that is what we're using to focus light. Our lens is pulled on by these muscles on either side. It's showing it on the top and the bottom, but really it's all the way around because this is a circular thing. Those are called the ciliary body. The ciliary body pulls on the lens and helps us focus. Behind the lens, we're now into the posterior chamber. The posterior chamber is filled with a gel called the vitreous humor, not the aqueous humor. Aqueous is liquid, vitreous is gel. So it's this big giant gel pad. And that gel pad helps our eyeball keep shape, but it also helps push the retina back where it's supposed to be, because the retina is not all that well attached. It actually can fall off. All around the rest of our eyeball, our entire eyeball, besides through the cornea, we have that white layer called the sclera. The sclera is thick, it's rubbery, it's protective. Then we have a layer called the choroid. The choroid is supportive. It's going to help us get everything we need into the cells of the retina which is the next layer. The retina is what you're going to use to see. Your retina is embedded with rods and cones. There's a place on your retina called the fovea centralis. That's where you have your best sight. So when you're looking at something, when you're reading words and you read this word, this word, this word, this word, what you're really doing is you're pointing your fovea at every word. So when you're looking at something, you're pointing your fovea at it. Your fovea is really your best sight. Everything else your brain kind of fills in. So here's your little fovea centralis. Then you have the area where your blood supply and your nerves go out. This is your blind spot. So you can't see anything where there isn't retina. And we have a blind spot in each eye. We just fill it in. Your brain's like, I can assume what is there. And so we make up what's there, but we actually can't see what's there. It's pretty incredible. Your eye is so connected to your brain that it's often referred to as an extension of the brain. That's because you are making a lot of assumptions and extrapolations based on the images that you're actually seeing. All of the information that goes to your brain is upside down and backwards, and your brain flips it and puts it right side up. And that's how we interpret everything. So what we actually see and what we think we see are not the same thing. All right, did I hit everything? I think so. Let's look at your eyeball again in a different orientation. Now we're going to talk about how light activates rods and cones. So here's my cornea, there's the space, there's my iris, here's the hole, then the lens, and now my giant gel pad. This image is showing a connecting line. That's supposed to be telling you that there's a blind spot here. It's not a physical structure. So it looks like your vitreous humor has a right and left half. It doesn't. It's continuous. This is just an area of darkness where you can't see. That's what it's trying to show you. Let's follow the path of light. Light is coming from somewhere. Right now I'm in my room, but it's dark outside. So the light's coming from the light bulb above me. It is traveling off of all these surfaces around me because light bounces. And it is bouncing into my cornea, through it, because it's clear, through the aqueous humor, through that hole in my iris called the pupil, through my lens, which is clear, because I have to be able to see through it, unless I have cataracts, and then it's not clear. Through all of that gel pad, through my vitreous humor, and it's going to hit my retina. It's going to hit the fovea if I'm looking at that thing, but it will hit my retina anywhere where my retina is, where it covers like the back 2/3 of your eyeball in all orientations, not just the right and left. When that light hits my retina, it's going to move through all these nerve fibers, through these neurons, which are associated with supporting your retina and helping you see and interpret things, all the way through them down to my rods and cones, which are the last layer of cells. In my rod, I'm going to be picking up photons. In my cones, I'll be picking up color. Those photons hit your rods and cones. The packets of photons will interact with chemicals in the membranes, and they will create a depolarization event. That depolarization event will then go out my optic nerve up to my brain, and my brain will interpret it as seeing something. So if I show you my cute mug where I have a picture of my cat on it, and then I have the other cat on the other side, your brain is seeing the color. It's seeing gray, it's seeing pink, it's seeing white, it's seeing gold for his eyes. But then you're also interpreting it. You're seeing the pattern of this. You're knowing that it's coming from me. So you're like, oh, Catherine's probably showing us her cats because she's obsessed with them. And yes, I am. But your brain is interpreting it and making all these assumptions about it. What if this was actually a picture of lots of little pictures that look together like a collage? You know, your brain is just making these assumptions. And most of the time it's right. It's just incredible that we're able to so successfully figure out what's going around us, what's going on around us with the information that we're given. I hope that was interesting. Email me anytime with questions. I know this is really complicated. We could have an entire term-long class just on these types of senses. So if you have any questions, please email me. I would love to talk about it. Have a great day and we'll chat soon.